Excellent. So thank you very much for the opportunity and thank you for your vote of confidence in me. So I'm your new friendly vice chair who will remain in office hopefully for 3 years. So it's extremely exciting to be here and be a part of this new enthusiastic and extremely cheerful executive board. So I put together a small introduction about myself so you know a little bit more what are my areas of expertise and what do I know about biology and genomes. So I'm coming from a country of Soviet Union, so I'm that young. So I was born in Latvia before it became independent, in a very small town outside of Riga, just by the coast. So I did my bachelor's on monitoring of endangered species of legumes. So, and after spending like half a year in the field and half a year in herbarium collections, I understood that's not my cup of tea. So I went over to something more applied and something more in the lab. So I was working with pathogenic fungi the majority of my scientific career. And in conjunction to that, I moved to Sweden to do my PhD. So I'm located in Uppsala, in beautiful city. If you haven't been there before, you should absolutely visit. So the PhD I've done in the group of molecular evolution under the guidance of Professor Siv Anderson. And so that's around that time I realized that the future lies in molecular biology and in bioinformatics. So the thesis of mine was mainly on taxonomy and species delineation in anamorphic or asexual fungi because it's a very large problem if you ever try to identify fungus. So a combination of molecular markers and the knowledge about the fungal biology was always needed. So I was doing DNA barcoding before it started to be called DNA barcoding, which was sort of cool. It was just in the early ages or early times of RNA operon sequencing and using COX-1 markers and so on. So I wanted to move over to the genomics and bioinformatics because I got extremely interested in that. So I started small with pathogenic bacteria of the genus Bartonella. So it was a lots of really advanced molecular biology and to look in the mechanisms of adaptation of the pathogenic organisms and look into the genomes. So why are they adapted and living in the human hosts? But then after that, I got my knowledge from my PhD. I got my knowledge from the first and second postdoc. And I decided to go back to fungi to apply the knowledge what I gained in my first postdoc towards studying genomics of fungal functions. So it was working on the most extremophilic organism on Earth, which is Zeromyces bisporus, that can go on honey. It can grow on your candy, but it's not harmful. It just spoils lots of really nice foods, including chocolate. So why sort of I want to bring in this slide because I heard a lot that people would like to know more about assembling genomes and bringing genomics in their lives. So I want to tell you all, the ones who are struggling, that I've been there. So I've been that person who did genome sequencing because at that time in the late 2000s, people just started sequence genomes to answer their biological questions. I was that person who got their genomes on the USB stick. I had no idea what to do with that, how to go from the genome that I've got from all these FASTA files into deciphering the function. So that was 15 years ago when we just started creating these infrastructures, and bioinformatics of the genomics was still in the very beginning. So I have this hands-on information, and I know how daunting it can be for a person who is either in the field or just in the lab to understand the inner workings of working with a non-model genome, with creating a new reference genome. So this is a little bit of the bingo. How do you know that you have been in the field of genomics for too long? So I've done isozymes. I don't know people who are smiling here. So you can raise your hands or smile at me. I see your faces. So I've done rapids. Remember what it is? That was a disaster. I've done slab gels for Sanger ABI on polyacrylamide. Well, and P20 or P32 labeling I've done during master's course. But yes, I was casting the slab gels for ABI Sanger. I've done PCR under oil. I've done southern blot and hybridization to verify assembly quality. That was before the fantastic assembly curation. So yeah, that's age of dinosaurs. Also, I've done DNA extractions in situ and pulsed field, like very sophisticated genome-wide RFLPs. That was also before people understood the necessity of creating high molecular weight DNA for non-model organisms. So with all this baggage, I came into the position where I'm at now. So I also sequenced my genomes on Celexa and on Illumina Genome Analyzer too. I did 454 jumping libraries. And I was involved with the de novo reference sequence of RNA using ABI Solid. And I didn't do the work myself. I produced the data. I did all the experimental setup, and the poor Henrik, who could not be here due to the illness, he was crying on my shoulder while assembling this data. So, but I remember the color space. It was a disaster. So don't try that at home. Also, for you who did bioinformatics, Mira, who remembers Mira? That's 454 assembler. I installed it in 64 bits hardware, and bioinformaticians will understand the pain because it was written for the 32-bit system. So it took me 2 months, but I took myself through it. So also do not try that at home. But in the modern history, then, after I've been through all that, after I sort of learned bioinformatics by scratch and just purely by doing, I understood the necessity of having a good infrastructure and help other people in need to produce their genome. So I went over to become a project manager at SciLife Lab Genomics, which is Swedish National Genomics Core, a part of a large national infrastructure. So, and since 2012, actually, I am in the field of genomics of non-models. So from 2013, I've been working with PacBio sequences and also trying to understand why they do not work for many of non-models. For you who do not work with vertebrates, you know how difficult that can be. So due to my work there, I was invited to be a part of the Vertebrate Genome Project Sample Prep Committee, published a couple of papers with them. Then I became involved in the Earth by Genome Project, and up to date, I'm in the Sample Collection Processing Subcommittee. That's work that's very much in connection with what we in ERGA are doing within SSP. I've been ERGA on the ERGA Council for, well, almost 3 years. So now I stepped down. And also I'm a part of the infrastructure grant in Sweden, where we're trying to set up a infrastructure to process a large scale non-model genome sequencing for the projects like ERGA and EBP. From 2022, I'm working at BG, and also for the last 2 years, I've been leading or co-leading together with Stefan Pettersson the planetary biology capability at SciLife Lab. And Jakob Höglund, who is present here, is one of my co-leads. So what I'm bringing to the table in terms of a vice chair of ERGA is more than a decade of the project management experience, but also most importantly is 20 years of genomics. And that sounds a little bit scary when you think about that, but I've seen the technologies come and go. I've seen technologies being extremely promising and then disappearing. So I pretty much have hands-on experience of working either with the data or the technology themselves. And that's genomics of non-models. I never done a human genome sequencing, although I know how to do that and how to analyze that. But what's fun in humans? So, and also 11 years of long-read sequencing and DNA extraction R&D. So in very many projects, I like to go extra mile and help people to understand why their samples work and why not. So, and with that, actually, that is it about myself. So if you have any questions, I will be happy to answer.